Hello and welcome. My name is Vijay Ramchandani, and today I'm going to present an overview on alcohol metabolism. I would like to acknowledge my colleague, Dr. Sundarya, for her help in developing these slides and materials. First things first, why should we even know about alcohol metabolism? Why is that important? As this picture depicts, there is a huge variability in the vulnerability to alcohol use disorder across the world. This makes one think, why are some predisposed to alcohol use disorder, and what might the effects of alcohol or the pharmacology of alcohol have to do with this? To understand this, we need to understand the processes involved in the clinical pharmacological processes. So the first one is pharmacodynamics, or what alcohol does to the body. This is a function of its pharmacological effects at the site of action and depends on the alcohol concentration or level, and more specifically in the brain or the target organ. This is determined by what the body does to alcohol or its pharmacokinetics, and that includes the processes of absorption, distribution, and elimination. This is hugely variable. As you can see in this example, this is data from a study where 12 healthy volunteers were given a single standard dose of oral alcohol, and breath alcohol levels were recorded as a function of time. And what you're seeing here on this graph are the blood alcohol concentrations on the y-axis as a function of time on the x-axis. And each of these lines or curves represents the data for an individual. As you can see, the peak alcohol concentration is extremely wide, ranging from 20 to 70 mg% between individuals. You can also see great variability in the overall shape or trajectory of this profile. So given this range, it is critical to understand the underlying sources of these processes of how alcohol is absorbed, distributed, and metabolized. And this in turn can inform us about the variability in alcohol's responses or effects, and how that drives vulnerability in the risk of alcohol use disorder. This is an overview of alcohol pharmacokinetics or metabolism processes. As you can see, there is absorption of alcohol through the gastrointestinal tract into the liver, which is the major site of metabolism, after distribution into total body water. It is interesting to note that alcohol reaches almost all organs via this body water distribution and then is eliminated unchanged in the kidneys and lung, what is not metabolized. We will touch upon all of these processes and their associated variability and end with a brief recap on sex differences in alcohol pharmacokinetics. Let us begin with how various factors associated with alcohol consumption affects its pharmacokinetics. What we drink matters. As you can see in this figure, what we are looking at are different blood alcohol curves obtained in individuals administered different types of alcohol in an experimental study. And again, you're looking at the mean alcohol concentrations on the y-axis and time on the x-axis. Each of these curves are different colors, represent the different types of alcohol beverages administered. And what you can see is that after consuming spirits, shown in red, the alcohol concentration, the peak alcohol concentration, was much higher compared to wine, shown in black, and beer, shown in blue. And so because of the concentration gradient, since alcohol in the form of spirits is more concentrated, it speeds up its absorption as well and can result in higher concentrations. How much one drinks and the total amount of alcohol is, of course, another important. This figure captures the data after consumption of different amounts of alcohol in healthy individuals. And again, you can see the blood alcohol concentrations versus time for 4 different doses corresponding to 1 drink, 2 drinks, 3 drinks, and 4 drinks of alcohol. And as you can see that there is a almost proportional increase in the peak breath alcohol concentration. The maximum concentration occurs typically between 30 to 60 minutes after consuming the drink. And then there is a almost linear decline in concentration. So what we drink and how much we drink matters and affects the absorption of alcohol. So how is alcohol then absorbed once it is consumed? And what happens to that alcohol? And what are the factors that drive that alcohol absorption? As I mentioned, alcohol is readily absorbed from the gastrointestinal tract. Absorption can start right in the oral cavity from the mouth as alcohol is being consumed, although that is a very small proportion of it. A smaller proportion in the stomach, but the major site of absorption for alcohol is in the small intestine, where about 75% of the ethanol is absorbed. And it is more rapid because small intestines have a large surface area for absorption. Once alcohol is absorbed, it distributes into body water and there is no plasma protein binding. The rate of absorption of alcohol is highly variable, and there are many factors that drive this variability. We already mentioned the dose or the amount people consume, the concentration of alcohol in the beverage, but also factors like food intake, gastric emptying, gastric metabolism, and these are the metabolism that occur in the stomach, and hepatic first pass, which is the metabolism that occurs in the liver, can also drive these processes. So how does alcohol level compare after IV administration compared to oral administration? And while IV administration or intravenous administration, where alcohol is infused directly into the veins, is not a natural way in which people consume their alcohol, it is used experimentally to understand its pharmacokinetics and response. And what you can see here is that when individuals were given IV alcohol, you get the higher peak shown here with the red arrow, compared to oral alcohol administration of the same dose when given in the fasted state shown with the blue arrow. And so you see this difference between oral and intravenous alcohol. That's not completely unexpected because when you give alcohol intravenously, it is going directly into the veins and you can measure it more rapidly. There is also food effects seen when alcohol is consumed orally. And so comparing the blue arrow curve with these lower curves at the bottom, those are the blood alcohol levels seen following the consumption of meals. So compared to the fasted state, you see lower blood alcohol levels after food. Food can delay gastric emptying time, which is the time that it takes for the alcohol and the food along with it to get from the stomach to the small intestine, which can then increase its metabolism and lower alcohol levels. But food can also speed up metabolism. And so to understand that better, we're looking here at data from another study where alcohol elimination rates were measured after IV administration in the fasted state and following a high fat, high carb, or high protein meal. And so the elimination rate shown here in the lighter gray bar for the fasted state is lower than the elimination rate seen after the fed state, and it seems to be similar between the high fat, high carb, and high protein conditions. And what this tells us is that in this case, since the alcohol is given intravenously, the effect that we see, this faster metabolism of alcohol, is purely attributed to metabolism, since again, this is IV, so there's no absorption effects. Another factor that can affect alcohol absorption, which I mentioned earlier, is gastric emptying, which is the time that it takes for alcohol and food as well to get from the stomach to the small intestine where it will be absorbed. And so a study was done in comparing alcohol metabolism in patients who had undergone bariatric surgery where gastric bypass procedures are conducted. And what you see again here is that patients after gastric bypass, shown in the dashed line, have a much higher alcohol level compared to controls shown in the solid line on this blood alcohol time curve. And so this seems to suggest that bypassing the stomach and getting the alcohol more rapidly to the small intestine can speed up its absorption and levels. Interestingly, it was noted that in patients who underwent this type of surgery, there was actually an increase in alcohol use and problems. And while the exact reason and mechanism for this is a bit unclear and being researched quite actively right now in the field, it does seem that faster delivery of alcohol to the small intestine results in much higher levels in the brain that can make alcohol more addictive, but also the alterations in the feeding peptides like ghrelin that are secreted by the stomach can contribute to higher absorption and higher risk for alcohol problems as well. So moving on to distribution, as I mentioned, alcohol is a small polar molecule and rapidly and readily distributes in all body water spaces. So the distribution to organs and tissues is really a function of its water content, but also depends on blood flow and tissue mass. And so higher the blood flow and higher the tissue mass, the greater the distribution. Alcohol passes rapidly through biological membranes, and contrary to what people think, it is actually insoluble in fat. The volume of distribution for alcohol is approximately equal to total body water, which is about 50 to 60% of body weight. And this is one of the factors that contributes to the sex differences because, as we know, women have a lower proportion of body water physiologically and anatomically compared to men. And this can drive some of the sex differences. And we're going to review that again. So moving on to metabolism, this is an overview of the main pathways of alcohol metabolism. On the left, you can see the so-called oxidative pathway, where alcohol is metabolized via two main enzyme processes. One is the ADH pathway, which involves alcohol dehydrogenase and aldehyde dehydrogenase enzymes. This occurs primarily in the liver. And there is also a non-ADH pathway, which comprises two enzymes, the cytochrome P450-2E1 and the catalase. And these seem to be more common in other organs like the brain. There also exists a non-oxidative pathway, which generates products such as fatty acid ethyl esters and phosphatidyl ethanol. And these products are usually excreted in the urine following their formation. And they can be used actually as markers of drinking, as they are present in the body for several hours to days after drinking ends. Looking a little bit more closely at the ADH pathway, there are two main steps in the pathway as shown on the left. The first step, alcohol is converted to acetaldehyde by the enzyme ADH or alcohol dehydrogenase. And in the second step, acetaldehyde is broken down into acetate by the ALDH or aldehyde dehydrogenase enzyme. The figure on the right, which I've shown you before, I want you now to focus on the decline in blood alcohol levels after the peak has occurred. And as you can see, it follows what looks like a linear decline with time. And that is because the ADH enzyme saturates at relatively low concentration, so equivalent to about maybe 2 to 3 drinks. And so once it reaches that level of saturation, it can only eliminate alcohol at its maximum rate. And that rate is approximately 10 grams an hour or 15 milligram percent per hour if you measure it in blood or breath alcohol concentrations. And this is equivalent to about 1 standard drink per hour. There is a good amount of variability here, but it is a reasonable estimate as an average reasonable estimate. So the acetaldehyde is the intermediate product that is formed from that first step and typically is rapidly eliminated by the enzyme aldehyde dehydrogenase to acetate. If, however, it accumulates in the blood because of an interaction or because of genetic factors, it can accumulate and cause many adverse effects such as headache, gastritis, nausea, and dizziness. And many of these symptoms are thought to be considered to be similar to what one feels with a hangover. There are important genetic factors in alcohol metabolism. As we know, almost 50% of the variation in alcohol use disorder is due to genetic factors. And the two best known enzymes that drive some of this genetic variation are the genes that drive alcohol metabolism. And what I mean by that is that there are genetic polymorphisms or mutations in these genes for the alcohol dehydrogenase and the aldehyde dehydrogenase enzymes. And so the three most studied and most relevant are the ADH1B and the ADH1C, which are two isoforms or different forms of the ADH enzyme, and also the ALDH2. And as it turns out, these polymorphisms, which involve a change in the sequence of amino acids in the protein, which are driven by individual differences in the genetic code, result in isozymes that have different efficiencies of breaking down alcohol and acetaldehyde. And also importantly, these polymorphisms have different frequencies in the different populations. As you can see here in this chart, while the white European American populations predominantly have the ADH1B star 1 form, the common variant, in Jewish populations, it is the star 2 variant, ADH1B star 2 variant, which is present in about 20% of individuals. And in Black African American populations, about 15% of them carry the ADH1B star 3 variation. Interestingly, in the Asian population, the predominant form of the enzyme and the gene that drives it is the ADH1B star 2. And then with regard to the ALDH2 shown in the two rightmost columns, while all other populations have predominantly the star 1 form, within the Asian population, about 30% of them carry the ALDH2 star 2 variant. So how does that impact alcohol metabolism and risk? With regard to these variants in the ADH, what we find is that genetic variation in these enzymes are associated with increased alcohol metabolic rates. While for ALDH2, there seems to be an increase in the acetaldehyde level because the enzyme that is formed is actually deficient in its ability to break down acetaldehyde. And so you get an accumulation of that and also a decrease in alcohol metabolic rates. And this is what is thought to be related to the acute alcohol flushing response, the nausea and the reddening of the face and the tachycardia, the increased heart rate that is seen in individuals who have this very characteristic response when they drink alcohol, and which is seen in about 30% of Asians. But beyond the effects on alcohol metabolism and this flushing response, there might also be long-term consequences. And in fact, studies have shown that individuals who carry the ADH1B and ALDH2 variants actually show a lower prevalence of alcohol use disorder, which is understandable given that alcohol produces this aversive response when people drink, this flushing response, and as a result, these individuals' drinking is much reduced and their risk of alcohol problems is also much reduced. But interestingly, what has been found is that individuals with the ADH1B variation in particular seem to show higher risk for cancer, and that is thought to be again related to the generation of acetaldehyde and other byproducts, and in particular risk for esophageal, gastric, pancreatic, and breast cancer seems to be increased. Finally, onto elimination. As I mentioned, the majority of the elimination, about 90+% of it, is by the liver, but there are minor pathways in which alcohol is eliminated unchanged, and that can range from 2 to 10%. The three routes in which that happens is the kidney, which can correspond to about 2 to 4%, in the lungs, which can correspond also about 2 to 4%, and through the skin, which represents 1 to 2%. And while these are minor pathways, they have actually been kind of relevant to our studies and our understanding because this measurement of unchanged alcohol has been used as a direct measure of how much people have been drinking, because you can use a device such as a breathalyzer that directly measures alcohol expired through the lungs and also through the transdermal or the skin route using these new wrist and ankle worn sensors. And this has really helped the field in better understanding alcohol use and directly measures how much someone has been drinking. I would like to end with a brief overview on sex differences in pharmacokinetics for alcohol, because I think this is one of the major sources of variability in the population and has a major impact on alcohol's effects as well as some of the pathology that results from alcohol drinking. Sex differences can manifest in absorption because of differences in gastric ADH activity in terms of metabolism in the stomach and the liver. Volume of distribution, as I mentioned, the body water composition is different between men and women, and so women having a lower proportion of body water per kilogram body weight will result in higher levels of alcohol when they drink standard amounts of alcohol. And in metabolism, differences in liver volume as well as liver enzyme activity, also differences due to hormones such as estrogen and growth hormone that regulate liver function as well. And this becomes important because the sex differences in these metabolism and absorption processes appear to result in higher levels of alcohol in women compared to men. And then these higher levels can translate into a higher risk for alcohol-related organ damage, which includes the liver and the brain. So in summary, what I hope we have done today is given you an overview of the pharmacokinetics of alcohol and an appreciation of this variability across individuals. As illustrated here in another study, data from 44 healthy men and women who were given standard doses of alcohol, each line representing an individual. In that study, you see this tremendous 3 to 4-fold variability in the time course. And so you can imagine this exposure and variability in exposure can have major consequences on behavior and response, and the consequences will be even more variable in individuals. And I think what we've also done is briefly reviewed with you these factors that can underlie this variability and how they might impact ultimately alcohol metabolism, its response, and risk for alcohol use disorder. And with that, I would like to thank you for your attention.